Finally, we'll be coming to the end of your syllabus, that is epigenetic regulation. Now, epigenetic regulation is a bit of a critical concept, but still to recapitulate the difference between genetics and epigenetics. When we are talking about genetics, if you remember the earlier day, the last day's class, I had said, let me have that. So the informations, we know that DNA has these sequences A, T, G, C organized in random spontaneous manner. And each of these sequences in sets of 3 form a codon, and each of a codon forms an amino acid, and amino acids are then garlanded in form of a string to form a polypeptide. So that is genetics. So the information present in the DNA moves to the RNA, and the information from some category of RNA, that is mRNA, can move to the protein. So the information from the DNA, if it moves to RNA, so DNA, we have the information moving from DNA to RNA, and the information then moving from some RNAs to protein. Not all RNAs are decoded into protein. You remember that only mRNA is decoded to protein. So the information moving from DNA to mRNA to protein, or the information can move from to other categories of RNA, which are known as non-coding RNA. It can be rRNA, it can be tRNA. And if you remember my first class, when I had given you the concept of a gene, that is a segment of DNA which is capable of producing a biological product. The product might be a non-coding RNA, that like the ribosomal RNA, the tRNA, or it might be, the product might be an mRNA, which gives a final product, protein. So segments of DNA which has protein as the biological product are referred to as structural genes, and the segment of DNA which has non-coding RNAs as their final product and referred to as functional genes. So it is this information pathway which is broadly referred to as genetics. But epigenetics is the study in which the modifications of the chromatin structure is done. See, modification of the chromatin structure is done in response to a variety of environmental factors. And these modification of the chromatin structure are reversible modification. So they can be reversed. So when the chromatin structure is modified in some manner, that forms the branch of epigenetics. So when we talk about DNA sequences and their inheritance, that is genetics. When we talk about the inheritance in the modification of chromatin structure, that branch is epigenetics. So this is a very broad, very lucid classification of what is genetics and what is epigenetics. Epigenetics is a very critical area and is a very vast area, but we will restrict ourselves to only 2 categories of modification of the chromatin. As you can see, one is the histone modification and the other is the methylation of DNA. We had touched some parts of DNA methylation in our previous classes. So when we come to this concept of epigenetics, there are epigenators. There are molecules which bring about this kind of modification. The molecules can be stress, it can be carcinogens, it can be UV radiations, it can be inflammations. Whatever that molecule might be, it binds with a receptor and starts a signaling process. Whenever a signaling process is started, that signal process is otherwise known as signal transduction. This signal finally reaches the proteins that will now bind with DNA to bring about their structural modification. So these binding proteins, or they can be small RNA also, These are non-coding RNA. So these binding proteins or small RNA are known as initiators of the epigenetic pathway. So in the epigenetic pathway, we have epigenators. These are signaling molecules that first start the process of signaling. The signal reaches the nucleus where we have initiators, DNA binding proteins and small RNA. These molecules then bind to the DNA and bring about certain structural changes in the pattern in which the chromatin is organized. These structural changes are reversible and hence, and these structural changes are brought about by actually activating certain category of enzymes. These enzymes are known as maintainers. These enzymes maintain the structural conformation of a DNA in how it inherited from its parent cell. So the inheritance pattern of the structure or the structural conformation is maintained. And this maintaining of the structural conformation is done by this category of enzymes known as maintainers. For maintaining the DNA methylation pattern, we have DNA methylases. For maintaining the histone modification pattern, we have histone acetylase. As the name suggests, it adds an acetyl group to a lysine amino acid. And histone deacetylase, again as the name suggests, it removes an acetyl group from the histone lysine residue. So this is the epigenome. So we have this epigenators passing the signal to initiators passing the signal such that the maintainers either change the conformation in favor of gene expression or change the conformation in absence of gene, in favor of gene silencing. So this is the epigenetic state of a cell. And this epigenetic state of a cell is known as the epigenome. We know our genome. A genome is the number of chromosomes present in a gamete, which includes our 22 autosomes plus X chromosome for female, and for male, the 22 autosome and X and Y chromosome, okay? But when we talk about epigenome, it is the maintenance of this state in the cell, which is known as the epigenome of a cell. Now let's move on to the two main categories of epigenetic modification. Now, we studied DNA methylation in the last day's class, but again, once more, this is a revision, recapitulation. See, cytosine is a common base in the DNA, and DNA methylase is the enzyme which adds methyl group to the 5 carbon, you can see. So it's 5-methyl cytosine. Now, the enzyme that is used is named as, you can see the name of the enzyme, DNA methyltransferase, DNMTs. As I said the earlier day, that cytosine is present in form of CG dinucleotides, and many promoter sequences have this CG dinucleotides present, which is also known as CPG. The CPG-rich segments are known as CPG islands. Now, why is this CPG islands a problem? The problem is that when methyl cytosine is present, it silences the DNA. If you add methyl group to the cytosine, the proteins cannot come and bind to the cytosine, to the DNA at that region. As a result of which, if the CG dinucleotide or the CPG island is present in a promoter element, the RNA polymerase will not be able to bind to the promoter element. So look at this picture. I've showed through this picture. Give me a second, this thing. I've showed through this picture. See, this is a gene, and this is the promoter element. And interestingly, in this promoter element has a CPG island. What happens? DNA methyl transferase is methylating the cytosine molecules. You see? So this is the chemical reaction in which the methylation has occurred, and it is methylating. So it is adding methyl groups to the 5' carbon of cytosine. And the moment methylation has been added to the CPG island, polymerase will not be. So if you remember RNA polymerase will not be able to come and sit on the promoter. So if polymerase is not being able to recognize the promoter, is not being able to sit on the promoter, then what happens? The gene was supposed to be transcribed. But since promoter methylation has occurred, the gene is now silenced. So this is the difference. No sequence has changed. Only a structural modification of cytosine has been done. Methylation has been added. Addition of methylation makes that area unrecognizable to the promoter polymerase enzyme, and the polymerase fails to recognize the promoter. And since the polymerase fails to recognize the promoter, the gene is automatically silenced. The gene cannot be transcribed. A beautiful example of methylation affecting gene expression is found in a category of disease known as the Fragile X syndrome, where in the gene is the FMR1 gene, and abnormal methylation of the gene in the promoter element completely silences the gene, and thus the females have an X chromosome which seems that it will break at any point. The X chromosome becomes very fragile in nature. So this is about DNA methylation. Now, this is more of a molecular picture to show you what methylation and histone acetylation can do. I have not gone to histone acetylation till now, but in my next slide, I'll be talking about histone acetylation. But prior to that, I want to show you that DNA can exist in two forms. One is the open configuration and one is the closed configuration. In the open configuration, you can see stretches of DNA are becoming accessible. This is the nucleosome model. So when DNA is in the open configuration, the DNA is partially opening up from the nucleosome so that it is being accessed to the transcription machinery. The RNA polymerase, if required, can access the DNA. Okay. But what happens? Methylation and histone deacetylation, keep it in mind. Methylation and histone deacetylation brings the DNA into a closed conformation. You see, as a result of methylation, these stretches of DNA are becoming inaccessible to the polymerase, and histone deacetylation is, you can see the nucleosomes are brought very close together. So we do not have long stretches of DNA where the polymerase becomes free to sit. So when we have DNA is unmethylated and histones are acetylated, the DNA is in open configuration. Contrary, when the DNA is methylated and histone deacetylated, the DNA is in closed configuration. In closed configuration, this is very vital. Genes cannot be transcribed, while in open configuration, you can see genes can be transcribed. So if DNA is in open configuration, it helps in transcription. If DNA is in closed configuration, it prevents transcription. So you see these are two modifications in the structural portion of the DNA that is actually allowing a gene to be transcribed or preventing a gene from transcription. We did the methylation part. Now let's move on to what happens when histone is acetylated and histone is deacetylated. What happens we are seeing here. When histone is acetylated, the DNA is in open configuration. When the histone is deacetylated, we are seeing that the DNA is moving into closed configuration. Now, what is this acetylation? What happens actually? So, I'll come to this picture here. Now, talking about histone acetylation, we all know that the DNA is wrapped around the histone molecules, okay? Now, so you have studied the nucleosomal model, so let's concentrate. So this is the core histone. You are aware of the core histone, okay? Now, these core histones often have an N-terminal tail protruding out, okay? So in this N-terminal, these are proteins. The core histones are nothing but proteins. So they have a N-terminal N and a C-terminal N. On the N-terminal N, there is this particular amino acid known as lysine. Lysine is a positively charged amino acid. Lysine in biochemistry is symbolized as K. Okay. So the core histones at the N-terminal end, they have this amino acid lysine. Now, when these enzymes, the first enzyme is the histone acetylase. The histone acetylase comes and adds an acetyl group to lysine. The moment you add an acetyl group is a negatively charged group, okay? So what this enzyme is doing, it is neutralizing the charge of lysine. Lysine is positively charged. Always remember that DNA is acid, deoxyribonucleic acid. So DNA is negatively charged. And histones are basic proteins. This is something you have read. Histones are positively charged. So it is this electrostatic reaction of positive and negative that helps the DNA to remain wrapped around the histone. So there is this electrostatic reaction. Now suddenly an enzyme comes and adds an acetyl group to one amino acid present in a histone. So histone gradually starts losing its positivity. Lysine, as I said, is positively charged. And when acetyl group is added to lysine, acetyl is itself negative, histone starts losing its positivity. The moment histone starts losing its positivity, this intramolecular positive-negative interaction starts to falter. Histone is gradually becoming neutral. So histone is now, as a result of addition of an acetyl group, the charge on histone is gradually becoming zero. So what will happen? Automatically, the DNA will have a tendency to open up from histone. Because this is simply an electrostatic interaction. It is not a permanent bond. DNA is wrapped around histone. It's simple electrostatic interaction. The moment the positive charge of histone is being lost, histone is becoming zero, DNA gradually starts opening up from histone. So you see, let's look now at this picture. This is the closed configuration of DNA. Just now we studied. And see, there is a promoter element present which is attached to which is present in the proximity of the nucleosome. Now, what happens? There is this recognition, a sequence for an activator molecule. If you remember, we studied enhancer. So activator protein comes and binds to an enhancer because we need the transcription of this gene. The moment the activator protein comes and binds to the enhancer, it recruits. We need to open up this part. Otherwise, if the DNA is placed in proximity to nucleosome, how is the RNA polymerase going to identify and bind to that promoter? So the DNA needs to open up. So what happens? The moment the activator proteins are recruited, histone acetylase enzymes are also recruited. So once histone acetylase enzymes are recruited, it starts adding acetyl groups to the lysine residues of histone molecules. The moment it starts adding acetyl groups to the lysine residues, the histone starts losing its positivity. And DNA being negative gradually starts unfolding from the histone because the positive negative reaction is hampered and histone has lost its positivity. And you can see from this picture, the promoter was inaccessible. Now you can see the promoter has opened up. So this portion of the DNA has opened up. This portion of the DNA can now be recognized by a polymerase, and now the polymerase can bring about transcription of this part of the DNA. After the transcription is over, what happens? Histone will not be in this open up configuration, because if histone key remains in this open up configuration, that particular gene will be constantly transcribed. I do not need huge amount of production of a particular product. So we need to fold back histone to that original configuration. So there we have a second category of enzyme known as histone deacetylase. So what happens? This histone deacetylase comes and removes all the histones that had been added. So once the transcription is over, once the product has been obtained, the activator protein opens up. Its job is finished. The moment the activator protein opens up from the enhancer sequences, histone deacetylase enzyme comes and removes all these acetyl groups. So if you remove all the acetyl groups from the lysine residues, what will happen? The lysine residues will regain its positive charge. Once the lysine residues again regains its positive charge, automatically DNA is negative, the histone has regained its positive charge, the DNA will again start folding properly around the nucleosome to form a closed configuration of the DNA.